The disclosures for guests on this podcast can be found in the show notes.
Hello, and welcome to the Journal of Clinical Oncology podcast, where we sit down with authors from some of the latest articles published in the Journal of Clinical Oncology. I am your host, Dr. Davide Soldato, medical oncologist at Ospedale San Martino in Genoa, Italy. Today, we are joined by JCO authors Dr. Sanjeev Kulgod, Chief Surgical Oncologist at Radnon Cancer Center in Hubballi, India, and Akash Kulgod, co-founder and CEO of Dognosis, a tech enterprise focused on the development of ultra-foldable, non-invasive breast-based multi-cancer detection tests. Today, we will be discussing the article titled Canine Olfaction Combined with Bayesian Modeling for Multi-Cancer Detection from Breast Samples, a phase 2 study in India. So thank you for speaking with us.
It's a pleasure to be here.
Thank you.
So I just want to start with the basics. We know that there is a real need for simple, acceptable, and effective cancer screening tests. And we know that there is a lot of difficulties into implementing this type of tests, especially in low resources settings. So I just want to set the stage by asking, what was the gap that you were trying to address with this study? And why did you believe that breath-based canine olfaction was a potential good test to test in this setting?
Yeah, I think we observed that there was a significant gap, especially in India, but broadly applicable to many countries in the global south and elsewhere, where there was no real way to stratify risk in a population such that resources for cancer screening could be targeted at those who needed it the most. In a country like India, not everyone can get a mammogram or HPV test or a pap smear. It really needs to be targeted if we were to cover the entire population of India to figure out where the people are who actually need further testing. And so this kind of enrichment felt like a real gap if we wanted to scale early cancer detection. And what we saw in the literature and I observed during the COVID pandemic and learned that dogs and what they're capable of doing with their sense of smell was something that we'd known for a while, to have some evidence that it was capable of detecting cancers and that it was also generalizable to achieve what is being called a multi-cancer early detection test, or MCET. And this was possible from breath, which is a modality that is not only easy to scale, because it's simply people breathing into a surgical face mask and can be done in any corner of a country like India without infrastructure or trained personnel. But it was also extremely acceptable to people. People did not have problems wearing a face mask because we've all worn a face mask during the pandemic, and it's something that's fully non-invasive. So it really felt like there was a missing gap and that we had the pieces present in the literature. It just required a study to really show that this was possible.
So you have introduced a little bit how this study worked, but this was a phase 2 study that wanted to show, let's say, a little bit of the efficacy of this solution and also the potential of including a different type of statistical analysis by including this Bayesian modeling. But I really wanted to break down the basics of the study. So how was the study designed? Who was included in the training and test cohort? And what was the primary question that you were trying to answer with this study?
It was designed to be an observational study or a case-controlled design, where we basically worked with clinicians in 6 hospitals in Karnataka in South India that represented both private hospitals that catered to middle and upper class from socioeconomic strata, as well as public hospitals and nonprofit hospitals, which catered to more rural and lower socioeconomic status individuals. And what we did was we collected samples from over 3,625 participants in total. And then we split it up in a training and a test data set. So the training samples were used to train the dogs and develop the system and the models. And then the test cohort were samples that the system and the dogs had never seen before until the day of testing. And during testing, we had a masking done for the handlers in the room who were calling out the indications. So they were blinded to what the samples were. And we had two sets, right? So we had individuals who had been confirmed to have cancer, and this was opportunistically collected. So it was any type of malignancy that was confirmed with a histopathology report. And in the controls, we had essentially both healthy attenders who were accompanying the patients or were present in the hospitals. But we also had individuals with other kinds of chronic diseases who were present in the hospital and were admitted for those chronic conditions. And we had a few individuals who had a benign report from their biopsy. So those were the controls, and we had the cases in the test split. And then in this assessor masked, blinded manner, we evaluated our system's ability to identify the individuals with cancers and their samples from the controls.
So basically, you had a training cohort that was used to train the dogs and train the system, and then a test cohort to validate these results. And this was really based on an analysis of breath. So really, in a practical way, what would you do? So people would breathe into the mask, and then how would you train and test then the dog to understand whether they were able to recognize or not recognize signals of cancer from volatiles that were contained in this breath?
Yeah, so I should have mentioned before, the sample here was simply a surgical face mask that an individual wore for 10 minutes. And during those 10 minutes, they were instructed to perform tidal breathing. So that face mask was then sealed in a medical grade Ziploc bag and then stored for up to 3 months at minus 20 degrees Celsius before being presented to the dogs. Now, during the training process, the dogs were essentially trained with positive reinforcement with quite a few different protocols and techniques that are present in the literature and that we also developed in-house. It leveraged a platform that we built that allowed for the reinforcement rewards as well as the logging of samples to be done by technology. And the platform had sensors and cameras that recorded other data variables as well. And then during testing, it was the same kind of platform that the dogs were then presented these novel samples from the test cohort that were then logged by handlers using software, where the results were not revealed to them until after the entire session and the test set was completed. And then we basically unblinded the samples and revealed the results.
And I think that, as you were mentioning before, we already had some data in the literature that showed that dogs were actually able to detect cancer. But I think that one very interesting point of the work that you have performed is that you have not relied on just one dog. So there were several dogs, and the masks, basically, they were evaluated by several dogs. So just wanted to break down the rationale for that for our listeners to understand why is it so important to have several dogs work on the same samples and to then integrate the signals from the dogs, because I think that that is a very important part of the work.
Yeah, you're absolutely right. And what we saw in the literature and also the reaction to the studies, right? Because you have over 50 peer reviewed blinded studies showing that dogs were capable of detecting diseases, including cancers, with quite high accuracies. But a criticism that was often levied was, how do you deal with the variability of an individual dog? What if the dog has a bad day? What if the dog, just doesn't perform during that session? If the result of your test depends on a dog who can be variable, how can this ever be standardized and then replicated across sites? And what we did was we basically developed a novel framework to integrate multiple dogs and their decisions using a Bayesian modeling framework. So what essentially happened was each sample was evaluated by at least 3 dogs, often as many as 7 dogs. And the indications of those dogs were either a binary yes, no in this study, but each indication was then tempered by that dog's historical performance. So the most sensitive dogs, the indication was upweighted, whereas the most specific dogs, their absence of an indication was upweighted. And what we did then for each sample was to say that you have the prior likelihood of that individual having cancer from their socio-demographic variables, their tobacco habits, their comorbidities. We'll take that prior. And then what we'll do is each dog provides a likelihood update that is weighted by their historical performance. And each dog's indications weighted in this manner will be aggregated to give an overall likelihood update that then results in a posterior score that we can then threshold using methods that are present in the literature to have a receiver operating curve. And this method of actually integrating multiple dogs by weighting them and then aggregating an overall score is, as far as we know, not been done in the literature of biomedical detection dogs. And what it actually does is quite profound, where we both get this quite stable accuracy and strong discriminative ability. Our area under the curve was 0.962. And for the threshold that was selected after calibration, we achieved a 91% sensitivity and specificity. But what also happened was you could simulate a dog having a bad day or that dog not being present for that sample at all. And you still had a stable area under the curve and you still had stable results for each sample because the model was able to incorporate the information necessary to give a standardized result.
One thing that is frequently brought up in this type of studies is whether the stage at presentation or whether the type of cancers, they might be detected in different ways. And so whether there is any type of things that we might not capture if we think about this being scaled up to a screening procedure. So did you do any kind of analysis to really see whether stage of diagnosis influenced the performances or whether performances were different according to cancer types that you identified in the study?
Yeah, so we did look at both cancer types and stages. We had an overall number of unique ICD codes of cancers in the test set were about 20. And we categorized them with the help of our clinicians into 7 broad categories. So these were head and neck cancers, breast cancers, lung and thoracic cancers, gynecological cancers, upper GI, lower GI, and then genitourinary. And what we really saw was, especially for 5 of the cancer groups, which had quite significant sample sizes, at least for the study, we saw stable sensitivity across the cancers. So it appeared that the dogs had generalized an overall scent of cancer that then was present in the volatiles in breath in sufficient quantities such that no matter what type of cancer it was, they were able to pick it up with the same level of accuracy overall. And this actually was also the case for stages. So we had about 100 of the cases out of the 283 in stage 1 and 2. And the sensitivity for stage 1 and 2 and stage 1 itself remained above 90%. So even though it was early stage, the volatiles in breath was still above the limit of detection of what the dogs were able to capture.
So basically, the model might be very robust when we look at this population, because as you mentioned before, even when you excluded one of the dog or when you simulated dogs having a bad day in terms of prediction, the model was still quite stable in terms of AUC for prediction, in terms of sensibility, specificity. There really doesn't seem to be any kind of differences when we look at cancer stage at presentation or different type of cancer. I just wanted to chat a little bit about how do you think that these results might be generalizable? Because I think that you made a very good job including a very diverse population, also including patients coming from the hospital, patients recruited from a more community setting. But I also think that this was quite a very specific population, maybe also to this region of India, because the rate of head and neck cancer, the amount of patient consuming tobacco in a chewing matter, which is really something that it's not that common in Western country. So just wanted to speak a little bit about this potential issue, which you acknowledge in the paper, and also how this issue might potentially guide you in future research.
We have a more of a head and neck cancer in the country because of lots of use of oral tobacco. If you look at actually the cancers, the patients who had large number of the cancer patients who had a head and neck cancers had a tobacco usage. But even in the controls, also there are people because a large population do chew a tobacco. So that way, cancer-specific detection was possible. But if you look at cancer subtypes of head cancers, we have a lots of other cancers which are being picked up very well by the dogs and a good sensitivity and specificity. So other subtypes also have a similar ability of the dogs to detect these cancers. So looking at the performance of that sensitivity, specificity of a detection, I think it's a pretty possible these other common cancers are globally pretty common. So I personally feel this is pretty utility across the globe.
Yeah. In terms of next steps for us, we know that, we also discussed it in the paper that there are limitations to case control and observational studies. The next step really is to show in a prospective study that the dogs are able to pick up signals in asymptomatic individuals where spectrum and other biases aren't present, and that those signals, when followed up, actually leads to confirmation of undiagnosed early stage cancer. And so the idea for us is to actually build upon this study to launch prospective trials in India. We'll ensure that the populations in these trials aren't just from the region in North Karnataka, where we conducted a lot of the recruitment, but are actually pan-India. We'd have sites across different states and different regions in India. And we will also plan to essentially then in those conditions compare how our test layered in the current community camps with the 3 types of organ screenings that the Indian government recommends: oral, breast, and cervical cancer. How layering a breath test that is multi-cancer can then enrich the number of cancers detected in the same community camps that are being run. So we'd have an active arm with our test and those 3 cancer screenings, and we'd have a comparison arm of just the standard of care screenings. And we'd really then have an objective and the endpoint of downstaging in the cohort that goes through the breath test along with standard of care. So this study is actually being planned and implementation would begin in the next few months. And I think that's the real test for this study, if whether it really can generalize to real world clinical utility. People have trained dogs across the world to detect cancers. At least 10 countries have launched such similar studies. So we know the signal exists and the dogs can pick it up. I think what's really been missing is this prospective validation and finding the right use case for what the dogs can do. We don't think this would replace standard of care or even compete with the liquid biopsies. We think this can really be a valuable enrichment layer and a risk stratification tool that could then enable all downstream diagnostics to work with a higher prevalence group.
Yes, but I also think that putting it in perspective, even just the model with the dogs alone, so without considering the prediction that was made based on the electronic health records, it already had some very good potential in terms of sensitivity, specificity, and AUC. So I know that there is a lot of fuss about liquid biopsy as potential early detection tools, but I also think that as we are looking for implementation, especially in low or middle economic region, I really think that this might be a strategy that is worth pursuing. And I think that the objective of really measuring this against what is standard of care in those regions, even though if it's potentially something that it's not widely applied in the West because we have better screening methods, I think that it really has a lot of value. So I think that this concludes our podcast for today. I just want to thank again Dr. Sanjeev Kulgod and Akash Kulgod for being with us today.
Thank you.
Thank you so much. It was great to be on.
So we appreciate you sharing more on your JCO articles titled Canine Olfaction Combined with Bayesian Modeling for Multi-Cancer Detection from Breath Samples, a phase 2 study in India. If you enjoy our show, please leave us a rating and review, and be sure to come back for another episode. You can find all ASCO shows at asco.org/podcast.
The purpose of this podcast is to educate and to inform. This is not a substitute for professional medical care and is not intended for use in the diagnosis or treatment of individual conditions. Guests on this podcast express their own opinions, experience, and conclusions. Guest statements on the podcast do not express the opinions of ASCO. The mention of any product, service, organization, activity, or therapy should not be construed as an ASCO endorsement.